Okay, hello and welcome everyone. Thank you for joining us today. My name is Tippy Duan and I'm with Stomis Global. I'm excited to talk to you about a new solution today, which is a spatial multi-omics platform that we call Stereo-CIT. Today we'll explore how this technology brings together the world of the transcriptome and proteome to give you an unparalleled insights into research. To ensure a smooth session, I'll be turning off my video recording right now. So as Stomix, we are bringing unparalleled insights to various applications at every scale. Our Stereo-CIT platform is a leader in the market and remains the highest resolution and largest field of view spatial single-cell transcriptomics platform available. The technology has been widely adopted by the scientific community with over 100 established Stereo-CIT labs and more than 220 research articles published using Stereo-CIT since 2022. So the power of our technology is rooted in our DNB-seq T10 system and the DNB-seq T1 chip. This platform utilizes a high-density DNA nanoball pattern array with coordinate information and allowing us to achieve a high resolution and a large field of view simultaneously. This means that we can capture and map the data at a resolution of 500 nanometers, giving you a subcellular details across an entire tissue section. So the outline of our workflow, which provides a complete tissue-to-data solution, is available for both fresh frozen and FFPE samples, followed by transcriptomics profiling and sequencing on our DNB-seq platform. We are also providing bioinformatics analysis suite, which includes StereoMap and the Stereo-seq analysis workflow to process the data, create a high-resolution spatial gene expression matrix, and visualize the results. This end-to-end solution is designed to guide you all the way from your biological sample to scientific discovery. Here we're broken down our various solutions by category, including the analyte to target, the compatible sample types, and the features. Today, we're focusing on the Stereo-CIT proteotranscriptomic solution. This solution is specifically designed for multi-omics studies, allowing for the co-analysis of both RNA and proteins from a fresh frozen sample, providing a more complete picture of your cell's biology. So let's discuss why adding spatial proteome data is so powerful. It helps strengthen our spatial transcriptomic insights in three key ways. So firstly, it significantly improves cell typing and clustering. Combining the modalities yields cleaner spatial domains and better separation of low-frequency populations. Second, it allows us to capture functional protein states and post-transcriptional regulation that RNA alone often misses. Proteins are the functional molecules. Identifying the spatially distinct distributions of the protein subtypes can reflect cellular functions that will be inferred less directly from transcripts. And finally, it enables us to perform more robust pressure analysis and biomarker discovery across different spatial niches within the tissue. In certain cases, proteins can be more robust biomarkers, and adding protein measurements increases statistical power to detect biologically relevant differences between regions. So these are just some of the examples highlighting how protein and RNA information can be used together to strengthen spatial insights. So in order to achieve this, Stomix have released a solution for protein and RNA co-analysis called Stereo-CIT, which stands for Stereo-CIT for cellular indexing of the transcriptomes and epitopes. It's a proteotranscriptomic solution that allows for the simultaneous characterization of hundreds of proteins along with expressed RNA. This slide shows clustering results from a mouse thymus sample, comparing clustering based on just the transcriptome, just the tag proteins, and a co-analysis of both. So you can see how the integrated analysis provides a much more refined and accurate picture, facilitating the cell phenotyping and revealing biologically relevant mechanisms. Stereo-CIT is a powerful platform with several key features. It enables spatial in situ multi-omics by providing unbiased spatial profiling of the whole transcriptome and hundreds of protein markers on the same tissue section. High-plex profiling within the context of intact tissue architecture is crucial for revealing information about cellular location and interaction that is lost in traditional methods. We also offer high resolution with single cell resolution for both transcriptome and protein analysis, facilitating precise cell type identification with multi-omics information to gain a deeper understanding of normal and disease biological function. And with high-plex protein detection, Stereo-CIT can achieve a high detection efficiency of over 100 proteins, which is far more efficient than traditional protein profiling methods. Finally, Stereo-CIT provides spatial protein profiling with high throughput sequencing, which avoids issues of autofluorescence and antigen instability that can be caused by multiple rounds of detection in other methods. The Stereo-CIT proteotranscriptomic solution is designed for unbiased whole transcriptome and protein analysis of fresh frozen human and mouse samples. It is particularly well suited for applications in immunology and oncology and can be used for standard-sized samples. The workflow involves determining optimal permeabilization time, followed by sample preparation, antibody incubation, imaging, and then sequencing on the DNBC platform. Because we're involving multiple antibodies in the same tissue section, we highly recommend users to perform a polystereocyte experiment on microscopic glass slides to determine the best concentration of the antibodies for you to use. So the Stereo-CIT solution uses commercially available antibodies sourced from BioLegend and Total-seq-A panels in combination with StoneMix Stereo-CIT proteotranscriptomics kit and reagent product. The principle of BioLegend Total-seq-A is to combine oligobar-coded antibodies with RNA sequencing to perform multi-omic profiling, simultaneously analyzing both protein expression on a cell surface and its transcriptome. The antibodies are conjugated to a unique oligonucleotide and allowing them to be captured by the poly-T probes on the Stereo-seq chips and then counted through next-generation sequencing alongside the poly-T captured RNA transcripts. BioLegend currently offers pre-mixed antibody cocktail panels or individual antibodies for you to build your own panels. The compatible species of these panels are human and mice. Many of the disease types and organs have been validated in-house. To give you a sense of the scale of the protein detection, on this slide, we're showing here some of the available panels from BioLegend. The mouse universal cocktail is designed to react with 119 unique cell surface antigens, while the human universal cocktail reacts with 154 unique cell surface antigens. So these panels are heavily focused on immunology and oncology markers, providing a powerful tool for profiling immune cell populations and their states. Also, spatial resolution is a critical factor achieving a comprehensive and a precise profiling of various markers, and Stereo-CIT excels in this area. So this slide, we visually compares the resolution of our Stereo-CIT prototranscriptomic solution to another platform, which is SPOT. Our nanoscale resolution allows for a much more detailed view of protein and RNA localization as seen in the mouse thymus and spleen samples. So we can perform both protein antibody-derived tag and protein immunofluorescence on the same section, and the added immunofluorescence doesn't influence the downstream protein sequencing while giving you a chance to validate the binding of the protein, the antibody and the protein on the comprehensive view of cellular architecture and molecular information. A critical question is whether adding this much of protein detection compromises the quality of the RNA data. As you can see from this mouse thymus sample, we're able to achieve the co-detection of a whole transcriptome with over 100 protein markers, and this does not significantly compromise the RNA capture efficiency. The number of genes and the sequencing saturations are comparable between the RNA only and the RNA with the protein experiments. This proves that our platform provides a high quality multi-omic data sets from a single sample. The comprehensive bioinformatics software suite we provide is also a key part of this end-to-end solution. The comprehensive software suite consists of three major components: the Stereo-seq analysis workflow or SAW pipeline, processes the raw RNA and the protein FASTQ data to generate a spatial RNA and spatial protein expression matrix and performs genome mapping and registration. StereoMap is a user-friendly desktop application for image processing and visualization. It allows for interactive visualization of marker gene expression and spatial clustering at single-cell resolution. In the case of Stereo-CIT, we have an example of the visualization capabilities of StereoMap showing the interactive display of both protein and marker gene information at the single cell or the cell beam level. This allows you to explore the relationships between gene and protein expression with spatial context. So now we'll dive a little deeper into some of the real-world application of using Stereo-CIT. So here we're profiling a human paracancerous lymph nodes. We perform a co-detection of the whole transcriptome and 163 proteins on the same tissue section. The images shown here on the spatial distribution of the whole transcriptome and multiplex proteins highlights the ability of Stereo-CIT to perform simultaneous multiplex protein and RNA detection without interfering with the RNA capturing efficiency. The colocalization of the four protein markers can be visualized with high fidelity as seen in the pseudocolor images on the bottom left, and the right panel shows a more detailed view of selected protein expressions. We wanted to demonstrate that the multi-omics joint spatial clustering can faithfully recapitulate the histological structure of a lymph node. Here you can see that the H&E image provides a morphological context, and the joint cluster analysis accurately identifies the key anatomical regions like the follicle and the germinal center. By comparing clustering from RNA alone and protein alone and the joint analysis, you can see that the integrated approach effectively distinguishes between cell populations that could be difficult to detect or separate with a single omic type, such as cluster 0 and 4 here and cluster 2 and 5 here. And you can compare it between the RNA protein UMAP with the RNA and protein joint analysis. The joint analysis further illustrates the contribution of each data type to the clustering results. So the plot shown in the middle are some of the cell populations, such as cancer-related B cells, endothelial cells, and a group of mast cells and platelets. They are better defined by protein information. While in contrast, other groups like B plasma cells and mast cells are more accurately defined by their RNA profile. So this highlights the complementary nature of RNA and protein data and how a joint analysis provides a more accurate and robust spatial clustering solution. We can take a further dive into how protein markers provide clearer spatial localization for certain cell groups. For example, for the cancer-related B cell groups, the protein markers such as CD20, shown here, and the PDL1, they show a much clearer and more localized expression pattern compared to the corresponding RNA markers. This demonstrates the value of protein information in precisely mapping cell types and states within a tissue. Besides identifying organizational structure and facilitate with spatial clustering using spatial multi-omics information, Stereo-CIT can also be applied to interrogate specific cellular activities happening at the invasive margin in human esophageal cancer. So first we have this image here. We have a pathologist identifying the invasive margin using H&E information obtained from an adjacent section. And we can see here that the spatial RNA and the protein expression differentiate from each other between the cancer and the dysplasia region, indicating specific cellular activities around the invasive margin. After annotating the cell types of different regions in the samples using the reference data set, we can see the epithelial cells are distributed we can see that the epithelial cells are distributed in the upper part, and the preliminary cell type distribution is consistent with the distribution of immune cells in the clear frontier of tumors in the early that has been demonstrated in early publications and literatures. We can observe that there is a large distribution of immune cells in the tumor area at the tumor invasion frontier. Here we can see the identified B cells and T cells and also macrophages with particularly high cellular activities around the invasive margin. Because immune cell types are typically less abundant and they exhibit lower RNA expression levels than other metabolically active cell types, they often show weaker RNA signals in single cell sequencing and spatial transcriptomics data sets. Therefore, when looking specifically at the immune activities, integrating protein information alongside RNA data is essential to achieve a more comprehensive and accurate understanding of cell identity and function. So in this esophageal cancer sample, we observed a high density of CD4 plus helper T cells within the tumor at the invasive margin, whereas the CD8 positive cytotoxic T cells were predominantly located in the adjacent normal tissue at the same frontier. So previous studies have reported that CD8 plus T cell infiltration within the tumor invasive frontier is generally associated with a more favorable prognosis. However, in our sample, the limited CD8 plus infiltration within the tumor region, together with the prominent PD-L1 expression at the invasive frontier, suggests the presence of substantial immunosuppression in the tumor microenvironment. Because of what we have observed and also in the field of cancer therapy, beyond assessing the overall immune status of tumor tissues, we wanted to look deeper into the heterogeneity of CD8 plus T cells. And this is critical for looking at the overall immune status. Particularly, we focus on examining the CD8 plus T cell exhaustion status, as previous studies have demonstrated that patients exhibiting an exhausted CD8 plus T cell subtypes prior to treatment tend to achieve a better therapeutic outcomes. So these findings underscore the potential predictive value of this exhaustion subtype for treatment efficacy, and we wanted to see how Stereo-CIT and the multi-omic solution, what are some of the insights we can provide. So in our analysis, we first identified the CD45 positive immune cell subtypes, specifically T cell populations, by integrating both RNA and protein information. The T cell status score revealed a spatial colocalization between cytotoxic and exhausted T cell states. Then leveraging the multi-omics data provided by Stereo-CIT, we further identify CD8 positive T cells within this population that exhibited features of exhaustion, particularly in clusters C1, C6, and C9, based on the canonical exhaustion marker receptors combined with histological context. So the exhausted CD8 plus T cell types, they were predominantly localized at the dysplasia region and along the invasive frontier. So this spatial colocalization of exhausted CD8 plus T cell subtypes at the tumor invasion frontier indicates an immunosuppressive microenvironment within the dysplastic area. Furthermore, when examining the CD8 positive T cells and their corresponding exhaustion ligand pairs within the local niche, we found that the ligand proteins such as PD1 and the TIGITL were significantly enriched around the CD8 positive T cell populations, suggesting that these cells were exposed to a pro-exhaustion microenvironmental pressure. Among the exhausted CD8 plus T cell subtypes, those co-localized within the tumor invasive frontier were most prominent. For further characterization of these subtypes, Stereo-CIT provides a comprehensive multidimensional data sets that can integrate transcriptomic and proteomic information with precise spatial context. So to help you choose the right platform for your research, here we are providing a comparison of our different solutions. We offer various transcriptomics and a multi-omics solution with different chip sizes, compatible staining methods, and species compatibility. So this allows you to select the best product for your specific research needs. And specifically for today, for looking into the multi-omic solutions, we have both Stereo-seq transcriptomics MRF and Stereo-CIT offering different methods and scales of detection. So here are some of the key differences between two of our multi-omics solutions, Stereo-CIT transcriptomics with multiplex immunofluorescence and Stereo-CIT. While both provide unbiased whole transcriptome and protein data, they differ in several key aspects. So Stereo-seq MIF uses microscopy-based fluorescent imaging for protein detection, which is limited to a low plex of 4 to 6 proteins. In contrast, Stereo-CIT uses a sequencing-based readout of antibody-derived tags, which allows for high-plex detection of over 100 proteins and provides a spatial gene expression matrix and a spatial protein expression data matrix for co-analysis. So Stereo-seq MIF is species agnostic, while Stereo-CIT is limited to human and mouse species since we are leveraging the protein panels provided by BioLegend. And hopefully, with the information provided here and some of the case studies we've shared previously, you will have a better understanding of some of the options we're offering for multi-omics platforms. Before I conclude, I want to highlight the resources available to you. You can access our global website for a wide range of materials, including software downloads, user manuals, videos, demo data, and FAQs. We encourage you to visit our website to explore these resources and learn more about our products that can support your research. Thank you for listening.